Firstly, what is gene therapy? Gene therapy is a medical technique that involves modifying a person's genes to treat or cure disease. The process works by addressing the underlying genetic issues causing the disease. How gene therapy work? Gene therapy work by delivering therapeutic or functional genes into a patient's cells. This innovative approach seeks to replace correct malfunctioning genes, offering potential cures or beneficial therapeutic outcomes. Gene therapies can work by several mechanisms, which is by replacing a disease-causing gene with a healthy copy of the gene, inactivating a disease-causing gene that is not functioning properly, and introducing a new or modified gene into the body to help treat a disease. Next is the delivery of gene therapy. There are two methods to deliver a gene therapy, which is by viral vector methods and non-viral vector methods. For viral vectors, it's utilize modified virus to transport therapeutic genes into cells. Common viral vectors include adenovirus, lentivirus, and retrovirus. These vectors are engineered to be non-pathogenic and carry the desired genetic payload. Viral vectors are highly efficient in delivering genes to the target cells due to their natural ability to infect cells. However, potential drawbacks include immunogenicity and the risk of unintended genomic integration. For non-viral vectors, in contrast, do not involve viral components. They include naked DNA, particle-based vectors, and chemical-based vectors. Naked DNA can be directly administered, while particle-based vectors use carriers like liposomes or nanoparticles. Chemical-based vectors involve synthetic compounds to deliver genes. Non-viral vectors are generally considered safer, with lower immunogenicity and a reduced risk of genomic integration. However, their efficiency in gene delivery may be lower compared to viral vectors. There are two types of gene therapy, which is in vivo gene therapy and ex vivo gene therapy. For in vivo gene therapy, therapeutic genes are directly delivered into the patient's body, aiming to treat the targeted cells or tissues within the organism. This method involves administering the therapeutic genetic material directly to the patient, allowing it to reach and express in the relevant cells. In vivo gene therapy is advantageous for its simplicity and the potential to treat a wide range of disease. However, it may face challenge related to targeted delivery and potential of target effects. For ex vivo gene therapy, it involves the extraction of cells from the patient's body, followed by the introduction or modification of the therapeutic genes in the isolated cells. After genetic manipulation, the modified cells are reintroduced into the patient's body. This method allows precise genetic modifications before the cells are returned to the organism, offering more control over the gene editing process. Ex vivo gene therapy is often used when direct in vivo delivery process challenge or risk.
What type of disease that we can use for gene therapy? The disease that may be treated by using gene therapy is hemophilia B. Hemophilia B is one ailment that may be handled in this manner. A defective gene that inhibits the generation of a protein required to coagulate blood following an injury is the cause of hemophilia. Hemophilia B genetic therapy entails injecting a vector with the normal clotting factor gene into the bloodstream. After entering liver cell, the vector produces the clotting factor required for improved health. Not other than hemophilia B, cancer also can be treated by using gene therapy. A wide range of inventive gene therapy has also been developed by cancer researchers, including specialized at Dana-Farber. Some refer to as ex vivo gene therapy, entail modifying the immune cells gene outside of the body and then refusing them. Others use viruses that have been genetically altered to combat cancer. Other than cancer, sickle cell also have used to treat by using the gene therapy method. Sickle cell disease can currently be cured by a bone marrow and blood transplant of stem cell from a related or unrelated well-matched donor. However, many patients lack access to a well-matched donor. For those without a suitable donor, genetic therapies that after an individual's own hematopoietic stem cell may be offered a cure. It is possible to inject altered hematopoietic stem cell into the bloodstream, and the cell will subsequently pass through the bone marrow space, and the cell can make healthy red blood cells that do not sickle once they are within the bone marrow. Although they are still relatively new as medical treatment, genetic therapy have the potential to treat a wide range of ailments. Some cancers, allergic reaction, and organ or tissue damage in the event of an injection are possible hazard. In many cases, genetic therapies are required to modify patient cells that are difficult to remove for laboratory therapy. The delivery mechanism for genetic therapy for certain ailments are currently being worked out by researchers. However, genetic medicine are now far safer and thanks to recent development. The FDA has approved several gene transfer medicine for clinical use in the US due to improve safety. Although genome editing is a far more recent technique than gene transfer, there have been a few clinical research on the subject. In conclusion, although genetic therapy are still in the early phase of development research and clinical trial, scientists have already learned a great deal about the potential therapeutic application for them. Genetic therapy could be employed in the future to treat, prevent, or cure inherited disease such as sickle cell disease, hemophilia, beta thalassemia, cystic fibrosis, and alpha-1 antitrypsin deficiency. They can also be used to treat HIV infection and cancer. For instance, we might receive treatment in one of two ways if you take part in a clinical study or eventually receive genetic therapy. Your body has the option of directly modifying your cell or of collecting, altering, and then returning your cell to you from outside your body. Your illness and the organ or cell type that require treatment may be determined the best course of action. That's all from us. Thank you.